Hello and welcome. In this lab session, we will discuss how could we use the online servers for the prediction of secondary structures of protein. At the end of this session, you will be able to learn how could you submit and analyze the secondary structure of protein by using online tool. How could you retrieve the information about alpha helices, beta strands, and coils, and how could you retrieve the information about the polar and non-polar amino acid residues. So in this lab session, we will use the Cyprid, which is an online tool for the prediction of the secondary structures of protein. So let's start with the overview of Cyprid. Cyprid is a protein structure prediction server that allows their user to submit their protein structures, perform the predictions of their choice, and receive the results via email and via on web. There are three methods which you can use by using the Cyprid, that is the Cyprid. You can select one of the three methods or these three methods at the same time as well. Cyprid is a highly accurate secondary structure prediction method. The other one is MEMSET2, which is a new version of widely used transmembrane topology prediction, and the other one is GenThreader, which is a fold recognition method, sequence profile based fold recognition method. So you can use these methods by using the Cyprid tool, and it is freely available for non-commercial users. So let's have a look on some practical example. To get into their home page, you just have to click on the above mentioned link. Then you will be redirected to their home page. As you can see over there, we have a lot of options. So among the options of select input data type, you can select the sequence data and you can select the PDB structure data. And these are those methods which we have discussed previously, like Cypred, MEMSET2, and GeneSetter. But for a time being, we are more interested for the prediction of secondary structure of our query sequence. So we will just select the Cypred. So in this way, just click on sequence data and on the Cypred. Uh, At the end of the page, you can also see a lot of other options which you can use if required. So you just have to put your query sequence into this box and you can write the name of the job, or you can write your personal email ID over here so that the link of the result will also be sent to you on the mentioned email. So you just have to then submit your query sequence. After submission, your query sequence has been submitted to the Cyprid tool. After some time, you will be redirected to your result page, and you can also receive the link on your email ID if mentioned. So this is the result page. As you can see over there that amino acids has been represented with a one letter code, and we have also a lot of options over there, like yellow color represents the strands, helices are represented with the pink color, and coils are represented with the gray color. So in this way, you can get the information about the secondary structure of protein. You can also save this information in the form of PNG and SVG. There is an option over there called Cyprid cartoon. If you just click on this Cyprid cartoon, you will be redirected to this result page. This is your query sequence which you have pasted previously. This is your query sequence, and as you can see that the pink color represents the helices, gray color represents the coil, and the yellow color represents the beta strands, as it is mentioned at the end of this page. Like yellow color represents the strand, pink color represents the helices, and gray color represents the coil. These are the confidence score, as mentioned over there, that this bar represents the confidence of prediction. Means जितना कलर यहाँ पे आपको डार्क नजर आएगा उतनी प्रिडिक्शन की एक्यूरेसी हाई होगी, और जितना आपको यहाँ पे कलर लाइटर नजर आएगा उतनी ही इस चीज की प्रिडिक्शन की एक्यूरेसी कम होगी, लाइक दिस अमाइनो एसिड रिप्रेजेंट्स द कॉइल. सो इन दिस वे आप इस बार के थ्रू यू कैन फाइंड आउट द accuracy of your prediction. Next one is, if you are more interested to find out how that which amino acid are polar and which amino acid are non-polar, you just have to click on the option show AA types. जब आप show AA types पे click करेंगे, you will be redirected to this page. आप इस पेज पे आ जाएंगे. यहाँ पे आप देख रहे हैं कि ऑरेंज कलर रिप्रेजेंट्स द नॉन पोलर small non-polar amino acid residues, green color represents the hydrophobic residues, red represents the polar, and the blue represents the aromatic plus cysteine. So you can also save this information in the form of PNG and SVG. So in this way, you can very easily find out or predict the secondary structures of your protein. You can get the information about which amino acid represents the coils, helixes, and beta strands, and about the polar and non-polar amino acid residues. Thank you very much.